Good morning. Welcome to the Saturday Science for Students at the Explorers Club. I'm Dr. John Tanacredi, and I am going to talk to you a little bit about this program. It's something we're actually celebrating our 10th year at the Explorers Club. The purpose of the program was to enlighten as much as possible high school students and college students about careers in the sciences and to understand how scientists get to where they are. So we've invited in colleagues and friends and collaborators and people who have the careers in a host of scientific disciplines, which are called STEM these days: science, technology, engineering, and mathematics. Also, it's involved with art, associated a collection of art programs and activities that were actually analyzed by scientists who looked at Leonardo da Vinci's work, and we brought in to make it STEAM as an important part of college and undergraduate and high school science training. The important thing about the Saturday Science for Students is that it's at the Explorers Club. It is the Explorers Club being the World Center for Exploration, and we like to say that the scientists and the explorers associated with the club have gone from inner space to outer space, from astronauts to oceanographers to people who have been involved with expeditions exploring all of the intricacies of natural systems. So one of the things that we want to bring up to the forefront here is new members of the club, students, can partake in activities that the club has gone on. So a little bit about what started 10 years ago as my own career, talking to students who were coming to the club. And we wanted to find out, I've been a member of the Explorers Club since 1988, and I was involved in something that's a very big part of the club, and that is called flag expeditions. There's a whole committee that brings the Explorers Club flag, and I wish you go to and hope you can go to the website, the Explorers Club website, and see the full range of activities and programs. Look into their literature, their newsletters, and the Explorers Guides that are very, very important and inspirational to young people if they're going into this career, science career. Just a little bit about my background and experiences and what prompted me to continue this program at the Explorers Club. I started my career, I'm born and bred in Brooklyn, New York, went to public schools in the city, got my Bachelor of Science degree in Biological Sciences from the City University of New York, and my master's degree from Hunter College Institute of Health Sciences, and my PhD is in environmental engineering it was Brooklyn Poly and Polytechnic University. It's now part of NYU Tandon School of Engineering. So one of the important things besides education and taking as many courses in the sciences and the science, engineering, mathematics, certainly is the idea of a network. Your participation in this Saturday Science for Students event is the beginning of your network. The concentration of knowledge spread out amongst many, many other people are really important for you to tap in as you go through your education and as you look toward a career if you're interested in science. I was a flight meteorologist with the United States Navy and flew was a part of the Hurricane Hunters, flew the 1969-1970 hurricane season, and the little bit of notoriety there in 1969 was Hurricane Camille, which was a level 5 hurricane and one of the largest hurricanes it was the largest, most intense hurricane to hit landfall, came ashore as a level 5, over 200 miles an hour, a 60-mile eye, and a 22-foot storm surge. So we're looking at, this is the end of the hurricane season this year, so put it in relative proportions, Camille was pretty intense, 1969. I was from the Navy, getting back into finishing up my education. I had a job. My first employment was with the U.S. Coast Guard as a bridge administrator, learning about environmental law. If you're interested in law, certainly you have to have grounding in the natural sciences and physical sciences, and it'll make you much better in dealing with the environmental regulation and and any good law gets implemented and enforced. So the idea of air and water pollution, all of these types of things was a big part of what I was being educated on in my first job, the U.S. Coast Guard Bridge Administration on Governor's Island in New York. And today you can visit Governor's Island. It has a portion of the National Park Service there. Then for the next 26 years, from 1978, I was a research ecologist with the National Park Service. And I worked on the Coastal Barrier Parks program and worked on World Heritage, where I had an opportunity to do 3 expeditions through the Explorers Club to Easter Island. And I have a book on Easter Island and our research that was dealing with over 20 scientists and a past president of the Explorers Club, John Lorette, who was inspirational to me in dealing with exploration and science in general. And John and I did these expeditions, and our book on Easter Island is a good introduction to World Heritage and the types of things that the National Park System does all the time. I retired from the National Park Service in 2000 and 2001, and went to Dowling College. I was the chairman of the Department of Earth and Marine Sciences, but it was at Dowling through a New York State collaborative. We established the Center for Environmental Research and Coastal Oceans Monitoring, CERCOM. So if you go onto our website from Malloy College and CERCOM, you will see its history. We're at the West Sayville Boat Basin in West Sayville, Long Island, New York. And we're a center for environmental research, but most of it is the M word, monitoring. We have collaborations with the National Weather Service. We have a weather station. We have a tide gauge with the USGS. We do work with the Fish and Wildlife Service, with the National Park Service, and we've done with water quality. And right now, we do the horseshoe crab monitoring, the 115 locations from the tip of Brooklyn to the tip of Montauk. I will leave you before my presentation, and if there's any questions at the end of the presentation, please hold on to them until then, and try to, each of the presentations will have time for a Q&A at the end. But I leave you with 3 important things that are necessary for all students, whether you're in high school or you're an undergraduate, you're just starting out in your career. The first thing is that building that network. Always you go to things that are free, but also go to things volunteer on a volunteer basis. I know that's difficult in these times. I'm looking for employment and opportunities in very, very difficult and unprecedented times. But volunteerism is most important. I started as a volunteer at the New York Zoological Society's New York Aquarium. I was a docent there and met the inspiration to my science career, which is Dr. George Ruggieri, who wrote a book called The Healing Sea. It was all about pharmaceuticals for marine organisms. If it wasn't for that volunteer experience, I probably would have taken a different path. I volunteered for the New York Academy of Sciences and worked on a 10-year program as a scientist when I was with the Park Service, dealing with that, with industrial ecology. And then, of course, I volunteer for the Explorers Club, and I co-chair the Conservation Committee, and I've been involved with the Cold Spring Harbor Whaling Museum as well. So, whether you're a college student or you're or an undergraduate, certainly on the college level, always look toward academic travel as a big part of the program. I've taken students with me to the Galapagos, to Easter Island. We've been around the world over the last 35 years. It's an important part of your career to have those experiences. They become very important in helping you in the career path, and certainly for employment. And with that, welcome to the Explorers Club. We're hoping that you'll be able to come visit the club at some point. Right now, that's due to COVID-19, a bit restricted. But once you have the clear opportunity to come into the city, into Manhattan, come by the club and take a look at the World Center for Exploration. Thank you so much, and I will get started with our presentation right now.
My name is Chester Zarnock. I am a professor at Baruch College and the Graduate Center at City University of New York. And so I'll be telling you a little bit about the work that I do and my path. I think you just got some really great advice from Professor Tanacredi regarding volunteerism, research experience, making contacts, and getting outside of New York to experience natural systems outside of New York, I think is important as well. But like Professor Tanacredi, I'm a New York City kid. I grew up in the Bronx and spent a lot of time fishing in City Island and Orchard Beach and just spending a lot of time around the water. And I think those experiences as a kid really sort of set me up for my career and my interest in environmental science and marine biology. So I ended up going to a small marine biology school on the east end of Long Island called Southampton College. And it was out there that I also sort of began my career volunteering. My very first internship was at the Jamaica Bay Wildlife Refuge in Broad Channel, Queens. And as you'll hear later today, 20 years later, I'm still working in Jamaica Bay at the Wildlife Refuge. So these kinds of experiences that you have in college or in high school can be the kinds of experiences that become really important in finding your career path. But anyway, when I was an undergraduate at Southampton College, I got very much interested in aquaculture. Aquaculture is the farming of fish and shellfish for food, just like agriculture, but we're talking about aquatic species. And at that point, I was really doing shellfish aquaculture. And shellfish aquaculture is a growing industry here in New York. There are private companies that are culturing mainly oysters all throughout Long Island, and aquaculture in general is the fastest growing sector of agriculture in the U.S. So it's a really important industry. But I spent time working with the East Hampton Town Shellfish Hatchery, whose job was to farm oysters, clams, and scallops, and restock the bay, restock Peconic Bay, and parts of Montauk. And at that point, I really started to understand how important it was to have clean water in order to support this industry. So things like harmful algae blooms, sewage pollution really was a major impediment to the growth of this industry. So I started to develop a sincere interest in water quality issues that are important to aquaculture. And you'll see that's became a thread in my career. After finishing my undergrad, I went back to the city and started a graduate program at the City University of New York at the Graduate Center, and was doing my research at Brooklyn College, where there was a really unique facility called AREAC, Aquatic Research and Environmental Assessment Center. And AREAC was an interesting place for me because of my interest in aquaculture. I was exposed to sort of the traditional methods of aquaculture, growing shellfish in oceans and bays and flow-through systems. But AREAC used a new kind of technology. They used a kind of technology called recirculating aquaculture systems. So these are large tanks, which you can see a picture of here, This is a 1,000-gallon tank. So these are large tanks where you can grow high concentrations or high biomass of fish in a very relatively small footprint, right? So this is an 8-foot circular tank, but you can grow 2,000 pounds of fish in this tank. So the idea is that when you have this small footprint and you can grow fish in these tanks, you can do it anywhere. And so we were growing thousands and thousands of pounds of fish on Flatbush Avenue, and we were able to set up tanks like this in Bed-Stuy and Coney Island. And so this was a really interesting technology for me because of the potential it had for feeding urban populations. But what ended up becoming a key piece to my career was understanding how we filter the water in these tanks. So these are recirculating or water reuse tanks. And so the water is being used over and over. It flows into the fish tank and then into a series of filters and then back into the fish tank. And in order to do that, you need to be able to clean the water. And the way in which we clean the water was using filters that had cultures of bacteria. And these cultures of bacteria removed some of the harmful nitrogen that the fish produced from their feeding and excretion. And that became a really important interest of mine. And at the time, I was really focused on fish production and shellfish production. But I became very much aware of nitrogen cycling in these kinds of aquaculture systems. And the kind of nitrogen cycling that was happening in these aquaculture systems is very similar to the kind of filtration and nitrogen cycling that has at wastewater treatment plants and that occurs in natural ecosystems like coastal ecosystems, lakes, rivers, even fields and agriculture. You have the similar kinds of microbes and processes that are happening in these natural systems that we are mimicking in aquaculture systems like these recirculating systems or wastewater treatment plants. So having that understanding of how these filters worked was applicable across a broad range of environmental issues. And again, that became sort of an important thread in my career. So as I completed my graduate degree, I spent lots of time working in this aquaculture lab, but I also did plenty of field work. And my field work was in New York City, in particular in Jamaica Bay. And what became clear to me was how important nutrient pollution was to shaping the structure and functioning of Jamaica Bay and other coastal ecosystems around New York City. And that became a sort of a passion and an interest of mine. And so just to give you a little bit of background, the nutrients going into our waterways are coming from wastewater treatment plants. More than half of all of the nitrogen going into our local waterways is coming from wastewater treatment plants. And nitrogen is a natural element. It's an important element for all life. And nitrogen going into ecosystems can initially be very productive and result in some positive impacts on ecosystems. However, if you have too much nitrogen going into a system, it can contribute to negative impacts on ecosystems. So we know that too much nitrogen loading leads to a problem called eutrophication, or we have too much organic matter in ecosystems, which ultimately leads to low oxygen events like hypoxia. Excess nitrogen also leads to harmful algal blooms, and we know that it's contributed to the loss of tidal wetlands and salt marshes and seagrass beds. And the waters around New York City, the Hudson River estuary, are the most heavily nitrogen-loaded ecosystems in the world. So it just gives you an idea. We have New York City residents use over 1 billion gallons of water per day, And that used water needs to get treated and released into our coastal ecosystem. And the treatment doesn't remove all of the nitrogen. So that nitrogen, we have 1 billion plus gallons of nitrogen effluent going into our waterways. And Jamaica Bay, if we separate out Jamaica Bay from the Hudson River estuary, it's one of the most highest nitrogen loaded systems. And to just to give you an idea of the numbers of what this means, is that Jamaica Bay is loading about 1,200 pounds of nitrogen per acre per year. So we're adding 1,200 pounds of nitrogen per acre in Jamaica Bay per year. And to put that in perspective, a corn farmer, a typical corn farmer, would apply 150 pounds of nitrogen to an acre of their farmland per year. So 150 pounds of nitrogen for a corn farm, but we're adding 1,200 pounds of nitrogen per acre in Jamaica Bay. So we're adding a very high amount of nitrogen, and this has, as I said, some negative impacts on the ecosystem. And so I can show you what that looks like. This is a picture that I took just a few years ago in Jamaica Bay, and this is my boat. And while we were doing some research in the bay, and the boat is sitting in 3 feet of water. So you can see this is the sand here, and then this is all seaweeds. And so these seaweeds grow excessively because they have access to essentially an unlimited amount of nitrogen. And so they're able to grow very significant, and obviously they cover the beach, they cover the water column, and that has negative impacts on the system. This is a picture of a different site in Jamaica Bay, and you can see all the seaweeds covering the beach. And this is July. And if you look way down the beach, you can see this is a car. Don't ask me how this got here, but that's a car, and I'm going to show you a close-up of this beach. But this is July with lots of seaweeds all over the beach. And then this is September, the same beach, but now I'm a little closer to that car, and you can see there's less seaweeds, but if you look closely along the beach, you'll see all these shells, and these shells are a clam called the soft shell clam, Maya arenaria, and these clams are all dead or dying. You can see all along the beach, all these clams, hundreds of thousands, all the way into the water column, all the way into, to the subtidal zone. You have lots of these dead clams, hundreds of thousands of these clams. And these all died because of the lack of oxygen in the sediment and the water, because of all the seaweed being decomposed. As this seaweed gets decomposed, those bacteria use up the oxygen in the water and the sediments, and that basically suffocates all these clams and results in this massive mortality event. And this kind of thing happens all the time. It happens every year, not always at this scale, but there are places throughout Jamaica Bay and New York City where we find these kinds of mortality events every single year. So what do we do about this issue? Well, we need to reduce the amount of nitrogen going into our ecosystems. And the New York City Department of Environmental Protection has done a nice job of that. And so this is one of the wastewater treatment plants in Jamaica Bay. And you can see from 2007 to 2012, they reduced the amount of nitrogen almost 50%. And they've invested literally billions of dollars in new technologies, building new wastewater treatment plants to reduce the amount of nitrogen going into our local ecosystems. And that's critical. And we need to continue to see those kinds of reductions, because we still have ecosystems that are challenged by the nutrient pollution coming from wastewater treatment plants. So we need to continue to invest. However, we're finding, at least the New York City Department of Environmental Protection is finding, that we're sort of plateauing in terms of the amount of nitrogen that we can remove at wastewater treatment plants in comparison to the investment in technologies. So in other words, as we continue to invest in wastewater treatment plants technologies, we're only making incremental improvements in the amount of nitrogen being removed, right? So the cost benefit analysis isn't necessarily in favor of investing in new technologies. So it's time to start thinking about alternative approaches. Yes, we need to continue to upgrade wastewater infrastructure, but what are the other approaches that we can be thinking about to address this nitrogen issue? And so if we just look at the ecosystem as a system that processes nitrogen, we find that there are a couple of pathways that nitrogen may have when it enters an ecosystem. It can be retained, so it can be assimilated by seaweeds and phytoplankton, fish, and so it can work its way up the food web and be sort of retained in an ecosystem. It recycles nitrogen, just like you and I recycle nitrogen as we eat and excrete. And so there's short-term pools and transformations of nitrogen. But the one that I'm really interested in is this issue of removal. So there's microbes. If you remember, we talked about the aquaculture systems. There's microbes that in the coastal ecosystem that can transform reactive forms of nitrogen into nitrogen gas and remove it from the ecosystem. And so that can be a really important pathway. So it's a way of getting the nitrogen out of the ecosystem. Another pathway could be nitrogen could get buried. But my colleagues and I have done some calculations, and we found that the natural systems like salt marshes in Jamaica Bay can remove about 17% of all of the nitrogen going into the bay. So that can be an important piece as we trying to address this nitrogen issue. And what we also know is that this process of removal, this nitrogen removal that happens in natural systems, is enhanced in certain kinds of habitats. So salt marshes, for example, and oyster reefs. Outside of New York City, you can have things like seagrass beds and mangroves. Those kinds of three-dimensional habitats create the environmental conditions that are just right for the microbes that turn reactive forms of nitrogen into nitrogen gas. And so this process, these microbes perform, is called denitrification. You know, that's a mouthful, but denitrification is the removal of reactive nitrogen into inert nitrogen, okay, or conversion of reactive nitrogen into inert nitrogen, which removes that nitrogen from the ecosystem. So from being dissolved in the water to being turned into a gas and unusable by the organisms in the water. And so that's a really important process. And the idea that these kinds of habitats like oyster reefs and salt marshes can enhance this process is really important. Unfortunately, we know that oyster reefs and salt marshes are also being lost. And so we've lost 80% of our oyster reefs worldwide. We've lost over 90% of our salt marshes within New York City. And so there are these habitats that are really important potentially in addressing this nitrogen pollution issue, but we're losing these habitats. So my colleagues and I have been studying this process for a number of years now, and we've demonstrated throughout our studies that these habitats, these three-dimensional habitats, provide this really important service of nitrogen removal. So what you're looking at here is this is a measure of nitrogen removal, and then three different habitat types. Subtidal sediments, intertidal sediments, and marsh. And you can see the marshes remove much more nitrogen than these two-dimensional habitats. I mean, this is a study that was funded by New York Sea Grant a number of years ago following Hurricane Sandy. So we we've been thinking about, this question of how to improve water quality, particularly in the context of the fact that we may be sort of reaching a plateau in wastewater treatment technology. And so we may be thinking more about how to remove nitrogen in situ within the ecosystem. And again, we can look at the different paths, right? And so if we think about the paths, the nitrogen can be buried, the nitrogen can be removed through denitrification, or the nitrogen can be sent out of the ecosystem through like flushing or hydrology. And so we've really been interested in this idea of ecological engineering, enhancing three-dimensional habitats. Like I said, places like New York City have lost a lot of their salt marshes, have lost their oyster reefs. And so what about rebuilding these, restoring these, in order to get these benefits of nitrogen removal and burial? And the idea, is something that we've been exploring for the last several decades. New York has New York city agencies and nonprofits have been working on restoration projects for a number of years. This is a restoration project that was in Jamaica Bay, a large-scale oyster restoration project. 50,000 oysters, 4 reefs, Billion Oyster Project, New York City Department of Environmental Protection, Hudson River Foundation, a number of different groups all getting together, trying to restore oyster habitat. And potentially this kind of habitat can be important in removing nitrogen. The catch is that somebody has to measure it, right? Somebody has to go out there and measure it to see if these kinds of restored habitats are actually providing the benefits that we think they might or could be providing. And so that's been a really important piece of my work. And this is some images of wetland restoration. And so this is a marsh in Jamaica Bay that was eroding. You can see it's all fragmented and falling apart, and was rebuilt. So they brought in sand and rebuilt this island, and then planted it with salt marsh plants so that it could be restored. And this was a large project, $20 million project. There've been a number of other projects of this scale that have been done around New York City and elsewhere. And these kind of projects are done for a number of reasons, a number of important services that these wetlands can provide. And I'm particularly interested in this idea of nutrient mitigation, like nitrogen removal and carbon sequestration. But again, somebody has to measure these benefits and services. And just because we construct it, does that mean that they're going to function the same way that natural marshes function? And what we've learned over time is that restored marshes can take years to decades to reach a functional equivalence of natural marshes. And so that's what this figure here is trying to point out, is that it can take a certain amount of time before functioning in restored marshes is equal to natural marshes. And then there's all kinds of questions about like, how do you design these things to have functional equivalence? Are there different approaches that we could use? What kind of plants should be there? Will animals use the habitat? And so there's all kinds of questions that we need to address. And my research has really been focused on this issue of nutrient mitigation. And so we want to measure, nitrogen removal in these restored marshes and oyster reefs. But we also want to be able to measure the carbon sequestration capacity of restored marshes. And so we know that tidal wetlands can sequester more carbon than tropical forests. It's called blue carbon. And so if we rebuild marshes, are we going to be able to regain this important service of carbon sequestration, which could help us in the fight against climate change? But again, we have to measure it. So this has essentially been the focus of my research program for the last several years. We're trying to understand how restored habitats can provide ecosystem services, and we are particularly interested in this problem in New York City, where there is a lot of nitrogen being introduced to the system, and we know that the environmental impacts of nitrogen can be detrimental to these restored habitats, right? So we want them to be able to grow and survive and provide benefits, but they are also likely to be challenged by these restored habitats. So we need to be able to explore that through that lens of eutrophic conditions. And then we want to know about, how do we design these things? Does design matter? What plants are there? What organisms are there? What's the role of species interactions in these processes? So a lot of my work has been in Jamaica Bay. We've been working there on tidal marshes since 2015. And one of the reasons why we think this is a really great system for studying salt marsh restoration is because the marshes are, there's a number of marshes that have been eroded. You can still see in this image, we have the footprint of salt marshes in Jamaica Bay, salt marsh islands in Jamaica Bay in purple in 1950. Okay, it's the footprint of the marshes in 1950. 50 years later, the footprint of those marshes is highlighted in green. And so you can see how some of these marshes have shrunk quite a bit over those decades. And what's neat is that there have been a number of large-scale restoration projects at some of these sites. And so those are highlighted here, 1 through 4, Big Egg, Elders East, Elders West, and Yellow Bar. So every couple of years, there was a new project of salt marsh restoration. And we thought this was cool because it allows us to look at sort of a chronosequence of marsh maturation and development. So rather than studying one site over a long period of time, we can study multiple sites simultaneously using kind of like a space for time substitution approach. So it's a way in which we can look at the maturation of the restored marshes through time in a small time, over a small time period. And of course, we compared it to natural marshes. We compared these restored marshes to natural marshes. So we had a marsh that was falling apart and being degraded and eroding away. And then we had a marsh that was more stable. So a positive control and a negative control. So I see there's some questions in the chat. I think I'm supposed to go through the presentation and then I'll get to the questions at the end. So what do we do? How do we study these marshes? And how do we study nitrogen cycling and carbon sequestration in these marshes? Well, we spend quite a bit of time out in the field, measuring plant traits, measuring carbon dioxide emissions. We like to take sediment cores to measure carbon sequestration. We also take cores that become sort of like a microcosm. We put a core into the marsh and pull it out intact. So it's a totally intact piece of the marsh. And then we bring it back to the laboratory where we can do studies on that microcosm. This is some more field pictures where we're doing manipulations on the marsh. Here we're adding rib mussels to the marsh, which I'll talk about later. We get lots of students involved in our work, everything from high school to undergraduate, graduate, and postgraduate people contributing to our projects. These are some high school students out in the marshes. Yep, in the lab working on plants, and there I am out in the field with a high school student from the Rockaways. We probably spend more time in the lab analyzing all these samples that we collect. And so here's a student working on sediment samples, and so these will end up getting analyzed for carbon in the sediment. And then this is a picture of the core setup. So like I said, we take these cores that are an intact piece of the marsh, we bring them to the laboratory, and we flow water through them to sort of simulate the natural environment. And so here you can see a core. Here we have water from the bay that we pump into the core, and then the water flows out of the core. So it's continuously flowing in and out. And what we do is we measure the nutrients and gases going in and the nutrient and gases coming out. And that tells us what the microbes and the plants are doing to the nitrogen. Are they removing the nitrogen or are they adding nitrogen? And we can do all these sort of neat experiments where we can actually tag the nitrogen using isotopes and follow their transformations. And so it's just some really neat stuff that we can do in these kinds of core studies. So I'll just tell you a little bit about what we found. And so this figure is showing nitrogen removal. And what we're looking at are the different marsh restorations in green. So the youngest restored marsh to the oldest restored marsh. And you can see that in general, there's a trend as you go from young to old, there's an increase in the amount of nitrogen removal. And so we thought this was neat. As the marshes get older, we have more nitrogen removal. And then we compared how much nitrogen was being removed in these restored marshes to natural marshes and other ecosystems. And we found that the restored marshes are removing about as much or more nitrogen than other marshes from other ecosystems. However, the natural marshes in Jamaica Bay remove much more nitrogen than the restored marshes. So even the oldest restored marsh, which was 12 years old or 13 years old at the time, removes less nitrogen than the natural marshes. So again, it goes back to this question is, will they be able to achieve the equivalence of a natural marsh? And how long will it take? We know it will take at least a dozen years or more than a dozen years to get to this point. But we don't know if they will ever achieve this functional equivalence. So it'll take further studies to determine that. And will these restored marshes last that long is sort of a secondary question. We've also measured carbon sequestration. And so these are some data that we collected from the Harlem River, from some restored marshes in the Harlem River. And these are just 4 different restored marshes and the amount of carbon that was sequestered. And what we found is the amount of carbon sequestered per acre in salt marshes in the Harlem River was more carbon that is sequestered in New York trees per acre or New York soils per acre. So we think this is an important idea, right? And so if we have 580 miles of shoreline around New York City. If we were able to turn our shorelines into salt marsh shorelines, we would be able to sequester a significant amount of carbon that will help with our fight against climate change. I showed a picture earlier of some of our students planting rib mussels in a restored marsh. And so I want to come back to that idea. We know that rib mussels, these guys here, they live in association with salt marsh plants, particularly the low marsh grass called Spartina alterniflora. And this is a mutualism. And so those of you that have taken biology classes, you probably are familiar with this idea of a mutualism. It's an interaction between two different species where both species have benefits. And so when the rib mussels live with the Spartina, there's benefits for both partners. The rib mussels get protected from the sun, they're protected from predators, they have a stable place to live, and in return, they provide nutrients to the salt marsh plants. They can reduce salinity stress in the soil and can prevent erosion of the salt marsh. And so they provide some important benefits. Each species provides benefits to each other. But what's interesting is that when we do salt marsh restoration projects, we add the plants, we add the Spartina, but we don't add the mussels. Okay, and so this is a picture, a couple of pictures of salt marsh restoration projects, and you can see we've planted these mussels, these very neat planted these Spartina, these very neat rows of them, but no mussels. And so our research has been interested in this idea of what if we added the mussels to these restored marshes? Would we get greater benefits? Would it provide a benefit to the plants? Would it result in greater ecosystem service of nitrogen removal and carbon sequestration? Will it make the marsh grasses, the restored marshes, more resilient to erosion and climate change? And so, over the last few years, my students and I and colleagues have done a few studies. And one of our first studies by a graduate student, Jennifer Zood, demonstrated that when you have plots with live mussels, so adding rib mussels to plots of Spartina grass, you double the amount of nitrogen removal. Okay, so again, we're looking at nitrogen removal. And then we have a control, just salt marsh plants. And this is just shells, not live mussels, just shells. And you can see there's no difference between mussel shells and plants. But when you have plants and mussels, live mussels, you get twice as much nitrogen removal. So we know when we add mussels to these restored marshes, we're getting this greater benefit of nitrogen removal. And just this week, I've spent quite a bit of time setting up experiments with my colleagues and students to look at how the rib mussels are going to impact the resilience of these restored marshes. Will they help the marshes grow? Will they help the marshes be more resilient to climate change? And so this is a couple of pictures. This is pictures from yesterday, actually, of a restored marsh in the Harlem River, where we added these rib mussel plots, right? And so you can see some of the marsh grass growing out of this plot. We added all of these mussels. And then here are some other plots that we're going to compare it to, where have no rib mussels. And the question, our hypothesis is that when you have the mussels, there's going to be greater fertilization. The mussels are going to excrete and add nutrients that's going to help these plants grow. And therefore, you'll have a more resilient shoreline. So we just set this experiment up yesterday, and we have plots in Harlem River and plots in Jamaica Bay. And we'll be studying this over the next 2 years to look at this interaction. And the idea would be, in the future, if we see a positive effect of mussels, we might want to consider adding mussels to our marsh restoration projects from the beginning in order to get the benefits that they provide, like nitrogen removal and resilience. And so this goes back to this idea of we're interested in this issue of nutrient pollution. We're interested in this idea that these three-dimensional habitats provide all of these important ecosystem benefits. We've lost these habitats, but now we're investing in bringing them back. So when we bring them back, let's make sure that we restore them and construct them in a way that provides the greatest benefits to humans and to the ecosystems. And that's the sort of the path that I'm on in terms of my research project program, and in terms of the kind of educational work that I do at Baruch College with undergraduates and high school students. So with that, I'll end here. I just want to thank the agencies that helped funded our work over the years, the Hudson River Foundation in particular, New York Water Resources Institute, New York Sea Grant. And then we're fortunate to work with some great partners, the National Park Service at Gateway, New York City Parks, Randall's Island Park Alliance, and New York Restoration Project. And I benefit from working with a great team of scientists pictured here, as well as many students, like I said, from high school to postgraduate, that have contributed to this work. So I'll stop here, and I know there were a few questions that came up in the chat. So I guess we can enter the question and answer period. I'm sorry. So I'm just going to go back to the beginning, and Tara asks, what's causing the increased concentration of nitrogen around New York City? And so the nitrogen is mainly coming from wastewater treatment plants. So like I mentioned, New York City folks use over 1 billion gallons of water every day, in our kitchen sinks and our toilets, and all of that water has to go to a wastewater treatment plant, and they treat it. But they can't remove all the nitrogen that's in that water. And so that nitrogen goes into the ecosystems when the treated water is released into the local ecosystems. So we need to be thinking about how we can invest in wastewater infrastructure so that we can continue to remove some of that nitrogen at the wastewater treatment plants. But as my work kind of suggests, there are ways in which we can enhance natural ecosystems to get the benefit of nitrogen removal as well. So thanks for the question, Tara. Ivana, thank you. I'm glad you think my work is interesting. I appreciate that. Jenna asked if the questions take place every Saturday. I think there's a few per semester. Oh, Regina responded. 8 times per year, 4 times each fall and 4 times in the spring. You reach out to circom@malloy.edu for more information. So Ivana asks about restoration projects along the Hudson River estuary. I've been working mainly in Jamaica Bay and the Harlem River. I've also worked in the East River on oyster restoration projects, and the Hudson River on oyster restoration projects. I don't have any active research projects right now in those areas, but that's something that we always keep in mind, and always looking for opportunities to expand our work in those areas. So if not right now, but hopefully in the future, we'll be working in these other places. Thanks for the question, Ivana. So I think that hits all the questions in the chat. Anybody have any other questions? Okay, so Tara asks, why do New York City waters have the most nitrogen loads? It's a combination of the concentration of people in New York City, and like I said, all the water that we use, and then I think it was a period of time where we didn't invest enough money into wastewater infrastructure, and because of that, we had lots of nitrogen going into the ecosystem. As I mentioned, New York City has done a really good job investing money into wastewater treatment, particularly over the last few decades, and even within the last 10 years, they've invested quite a bit of money. But if you think about investing in wastewater infrastructure isn't the most compelling thing for most New York City folks, right? If you you ask a typical New Yorker, do they want their tax dollars going to repairing the BQE expressway, the Brooklyn Queens expressway, or wastewater infrastructure? They say, well, I'm stuck in traffic on the BQE every day. I want you to repair the BQE. But they don't think about where their wastewater goes. And so I think that's important. I think it's an important part of events like this to get the word out, to get people to understand the importance of wastewater infrastructure. And I think it's really important for people around New York City, Long Island, to be engaged with their waterfronts, right? I think New York City folks tend to not realize that they're surrounded by water. 580 miles of shoreline, right? 4 to 5 boroughs being islands. We're a port city, and it's really a tremendous resource. And I think it's important for people to get engaged and to get involved. There's questions about volunteering, to get involved with the groups that are doing work around the waterfront. Billion Oyster Project, Randall's Island Park Alliance, New York Restoration Project, Rockaway Initiative for Sustainability and Equity. There's so many nonprofit groups, organizations that are involved, waterfront issues that students can get involved with, that are really important in being stewards for our natural systems, for our coastlines. And so I think it'd be great to get involved with those groups. I think it's important to get involved with academic institutions like CUNY, like Malloy, so that you can get involved with the research that's happening around New York, because there's lots of really important work that's happening, and I think there's lots of opportunities for students to get involved in. So I think that hits Justin's question. And Jenna, I appreciate that you like the lecture. Any other questions from folks?
Well, Chester, thank you so much. Really appreciate taking the time today. And this presentation is recorded, so anyone who, if you pass the message about the Saturday Science for Students at the Explorers Club, you can retrieve this in video form. It'll be online at the club and also at CERCOM at Malloy College.